Chapter 3. Um, Chapter 3 is about the cells. And the first thing here is a reminder of the key concepts that we already covered, and then we're going to add other things. So we're going to talk about the cell theory, what's prokaryote versus eukaryote, and um, cells are small for a reason. We'll talk about it, plasma membrane, what does it mean, and then we will talk about the organelles. Okay, so what is the cell? We talked about it before. It's the basic unit of life. Anything less than that is part of the cells, but we do not call it basic unit of life. You have to memorize these things. It's an organized unit that can live, reproduce, response, right? Sense and response. That was the definition before. And you can utilize raw material to get energy, and you have DNA for instructions. What's the total number of cells in the human body? It is trillions. You need to remember this. Everything that I'm focusing on is important. Cell theory, I already talked about it before, but here it is again because you will be asked about it. It's important. What's a cell theory? Every organism is composed of one or more cells. For humans, we are multicellular. We have trillions. About bacteria, one cell. How about in between? Yes, archaea and others. There are in between. So this is number one. You have to memorize this and remember it. Cell is the smallest unit of life that have the properties of life, and that's why we call it the smallest unit of life. All cells come from pre-existing cells. Your skin cell fall down, die, and then another cell that you have, like stem cells inside your body or at the bottom of your skin, is going to create another one. Liver cell make liver cell, and so on. So cells come from pre-existing cells. So when we talk about the cells, we will talk about it this way. First of all, there is a membrane from outside. Okay, I usually try I always think about the cells as a factory. Like imagine this is a factory, this is the wall of the factory, and this is plasma membrane. It's called a plasma membrane or the cell membrane. And you need to remember, it's not called wall. It's completely different. Humans do not have a wall. Something like bacteria, they have a wall outside of this. It's not a wall. It is membrane. Plasma membrane, cell membrane, that's okay. But you have to say membrane. And this is like the outer wall surrounding the factory. The nucleus inside here containing DNA, I consider it like the manager's office in the middle of this factory, right? So this is the nucleus that contain our DNA. Why do we have the DNA? Because of the genes. It carry the instructions for everything. The manager directs the factory on all aspects. You're going to do this, you're going to do that. I have instructions for everything. What are the instructions? DNA. Cytoplasm is what's in between, nucleus and the wall, which is everything else in that factory. So the whole space here is called cytoplasm. Cytoplasm. What's cytoplasm? Let me tell you this as we go. Every time I'll give you more suffixes and prefixes, prefixes and suffixes. If you collect it together, that's going to be extremely helpful. Trust me. You will see it later as we go. Because Greek and Latin is the language of biology, anatomy, and physiology, and others. So it's completely different than English. Okay? How? Because here you have prefixes and suffixes, and you mix them somehow, put them together somehow, it will give you a word. So it's not every word is completely standalone or every word is unique. No, that's not true here. You repeat all these prefixes and suffixes, you put it together. Let me give you an example here. I'm going to cut this word into two pieces. Cytoplasm. Cyto, this is Greek for cell. Plasm is Greek for liquid, the liquid inside or the space inside. So what's the cytoplasm? It's the space inside the cells. Is that the only time that we're going to use these prefixes and suffixes? Actually, no. You will see it a lot. You will see here is cytoplasm. You will see something like cytology, cytoplasmic reticulum, cyto. You will see it a lot of times. It's the same word. And this is what's good about Greek and Latin is it's 2-3 pieces, you put them together. And if you know the pieces, even if you don't know the word, you can actually guess it and figure it out, even if you don't know. So this is for cytoplasm. So it's between the membrane and the DNA within the nucleus. The fluid part of it is called cytosol. So plasm is that space. Sol is the fluid part that's here. So what's the cytoplasm? What's the difference? You see all these brown things here? These are contents. And look at the liquid, the fluid, the blue. So the fluid alone is the cytosol. Cytoplasm is the whole thing surrounding the nucleus, everything, including the cytosol and all these organelles. As usual, feel free to stop me anytime you want, okay? If there's something that doesn't make sense or you want me to repeat, I want you to get as much as possible of this as we go. Prokaryotes and eukaryotes. I talked about that before, and I told you we need to cut it into two pieces. Pro, it's a prefix that means before something or not before the development of something. So pro and karyote is a suffix that means referring to the nucleus. So prokaryote, it's before it is advanced enough to get a nucleus. Before nucleus, no nucleus meaning. It means no nucleus. U means normal or complete. So this is the one that has the nucleus. Prokaryote includes bacteria and eukarya. Okay, so does the bacteria and prokarya have a nucleus? No, they do not. Do they have DNA? Yes, both of them do, but it is not included within the nucleus. As if the manager doesn't have an office. He's just moving around all the time. Versus eukaryotes. In eukaryotes, you will have a nucleus, and this is the most important one, that contain the DNA. Okay? There is a plasma membrane surrounding it, which is not much different than here. So both of them have a plasma membrane. And the one difference so far is both of them have a plasma membrane. Both of them have nucleus. What's the difference? I'm sorry, take it back. Both of them have DNA, I mean. Both of them have plasma membrane. Both of them have DNA. What's the difference? The DNA is in the nucleus here. It's not in the nucleus here. And it also contains membrane-bound organelles. What do you mean by membrane-bound organelles? These organelles, look at these. We will talk about it in detail. Look at all these brown. These are called organelles. And what's organelles? Organ-elles. Organ, like liver, like spleen. This is organ. If you put E-L-L-E at the end, it means tiny. So organelle are these tiny organs within like you have your organs, liver, spleen, heart. This is an organ. The cell have their own organ, very tiny organelles, like Golgi apparatus and mitochondria and lysosomes and so on. We will talk about it. Are our cells small or large? It should be small. It is small. Why? Because large cells cannot survive. It's microscopic. Micro, I think you know what's micro, very tiny. Scopic is scope, like the scope is too small. You can see it under the microscope. Macro is the opposite of this. So I'm giving you extra prefixes and suffixes here. Micro versus macro. So if we say macroscopic, this is for the big things. 2,000 red blood cells, this is how small it is. You can line it up across your thumbnail, believe it or not. If you put some blood like on your thumbnail, and I'm asking you to, can you tell me approximately? It's like 2,000. This is how small it is. And why it has to be small? Because this will give you large surface area compared to the volume. Large surface to the volume ratio. It's very large. If the cells are large, it will be the opposite. It will be small surface to volume ratio, and this is bad because you need as much as possible of the surface area as compared to the volume, so that every cell have its own in contact with the surrounding and it can do the exchange. The bigger, the less exchange, the less survival. So large cells cannot survive. Our cells are all small. So because of that, if you have enough surface area, you will not be able to do the exchange. What's the exchange? You take what you need in across the membrane and get rid of what you don't need, which is the waste product. You kick it out. You take the nutrients, you take the oxygen, you take the water in, and then you use it. And then the waste products, you move it out. You are free to do that thanks to the large surface to volume ratio. If it is not small, if we do have some cells that are not that small, but if that's the case, it's going to be long and thin, so that you can still increase the surface area, and some of them will be able to fold, and we will talk about that in details. If it is bigger, it's going to make folding in order to make the surface bigger with a smaller volume. Okay, if the volume is like this, and when you fold it, it will take less volume compared to the surface.
I'm a little confused. So does it mean that like the cells are sort of like flattened in terms of like they're more flattened and like disc-like compared to like I just don't think I understand.
Okay, it is like if you have a box that this is big, and the inside of this box is trying to communicate with the outside, and it's full of things. It cannot communicate well because it's too big. But if I make it into smaller pieces, it will have larger surface area. If you cut the box into smaller boxes, the surface area and so the interaction from inside to the outside is going to be bigger. If the cell is small, which is the case in our case, the cells are small. So the surface to volume ratio is large. And let me say it this way. You know what happened if I put those 2,000 cells as one cell, one big cell? The diameter, which gave us the surface, is a lot smaller than each one of those individually. Let's say just, I gave you a random example, okay? If it is 2,000 red blood cells, and let's say the diameter of each one of those is 1 micron. So what's the combined diameter of those, which is the surface area, that I mean to represent the surface area, it's 2,000. If I merge all of these and make one big red blood cells, it is not 2,000, it's going to be like 500. So is it the same thing? It is not, because now the surface compared to the volume is different. So that's why you take it apart, and the surface area is going to be bigger. So 2,000 cells, what's the surface area? 2,000. If you merge it in one big cell, red blood cell, what's the surface area? Like 200, 300, which is a lot smaller. So the more you divide it, the bigger surface you have. Does that make sense?
Can I ask you a question?
Sure.
So trying to understand it, are you saying that by dividing it into little pieces, do those little individual little pieces have the same information as a large piece would? So if you had a large piece, all the information that's in that large piece is inside the little piece. So you're actually turning all those little pieces into the same volume space?
What I'm doing here is these individual cells. I'm not talking about the contents. Contents is the same. Let's say I have
Okay.
2,000 red blood cells. Okay. Each one of those will have its surface area. If I mathematically, I didn't merge them, I just mathematically add up the surface area of these 2,000 individual red blood cells, it will give me a number. Let's say that's 2,000, the surface area. If I merge them, I didn't lose anything. The contents are the same, but instead of putting it into smaller cells, I put it in a huge cell. The surface area is not the same. It's going to be a lot smaller as compared to the surface area of the 2,000 added up together. So the more you divide it into smaller pieces, the more surface you have, not the contents. Contents are the same. Does it make sense now?
It makes a bit more sense now, yeah.
Is that because by it being little pieces, there's more space in between the little pieces that causes it
This
to
is
have more?
this is one, but the other thing is it is actually larger by counting. If I give you 5, let's say, let's do it this way. If I give you 5 soccer balls and I ask you to measure the surface for me with the air inside, and you say, okay, I did that. And then if I merge those 5 together, it's not going to be the same. It will be the surface area, the surface around, the surface area is going to be smaller when it is combined. So the more you divide it into pieces, the larger is the surface area so that they can interact with the surrounding. The other part is what you just said, which is more exposure to the surrounding. Okay,
Okay, thank you.
you're welcome. Here it is, it's giving you with numbers. If the width of a cube is 1, surface area is 6. If the width is 2, surface area is 24. And the comparison here, if it is 1, it's going to give you 6. The surface area is going to be, compared to the volume, is going to be a lot larger. You don't have to know these numbers. It's just explaining the same thing that I just talked about. Okay. And here it is again, it's giving you the volume is the 3 length, width, and height. This is a cube, so all of them are going to be the same. So if you multiply the 3 together, that's going to give you the volume. Okay. Again, that's not the most important thing. It's just explaining how the same concept, which is cells are small so that the surface area is bigger. Meaning surface area to volume is bigger so that you have more interaction with the surrounding. This is a bottom line. These are different types of cells that we're going to discuss. This is how the skeletal cells looks like. Look at the neural cells. This is big, not as small as the other ones, but it's long and cylinder to have more interaction and more interaction with the surroundings. This one here is a fat cell. This is how it looks like. If you look closer, this is how it looks like. This is how the liver cells looks like. If you look at the skeletal muscle cells, it's long and cylinder like this. And let me just, we are not going to go deeper into this, but let me just tell you what skeletal muscles mean. Skeletal muscles are the muscles that are attached to your skeleton. Like these muscles here, like here, this is my biceps, my triceps, attached to the skeleton, and it is voluntary. The muscles that you can move, like I'm going to bend my arm, I'm using the skeletal muscles. So the characteristics here is it's long and cylinder. The longest cell of the body is the neural cells. Okay, here is a nerve. This is the longest nerve of the body. I don't know if you guys heard about sciatica before, which is inflammation of this nerve, and the patient will have pain shooting down from here all the way to the foot. He would say the back of my buttocks, of my thigh, of my leg, even all the way down at my foot, all of them, the sole of the foot, all of this hurt me. So this is called sciatica. Maybe something pushing on it, maybe it's inflamed or something, but the nerves are the longest of all. Fat cells will look different than others. What's the fat cells? Yes, some of our cells, their job is to store extra fat. And because of the fat filling, literally filling the space inside, filling the cytoplasm, it's going to push on the nucleus and push it to the side, and it will be looking like this. If you look under the microscope on one of these fat cells, look, where is the nucleus? Compared to the other nucleus, look at this nucleus, for example, it's almost in the middle here, right? Other cells, let me show you other cells like this. Look at the nucleus, it's in the middle. Fat cells specifically, the nucleus is always at the border. Why? Because it is, the fat is being stored here. It's filling all the space or most of it and pushing the nucleus to the side. This is how it looks like. Liver cells, just a normal cell. Like it doesn't have to be exactly in the middle, but not pushed to the periphery. So here is the cell theory. Again, this is like the third or the fourth time to see it. Obviously, it's extremely important. What do you guys think? Every organism is composed of? Does anybody know?
One or more.
Yeah, one or more. What?
Cells?
Cells, yes. Okay, the cells is
Smallest.
basic. Smallest and basic, yes. Smallest, basic. All cells come from?
Pre-existing.
Pre-existing, yes. Very good, very good.
I had a question about like with the fat cells too. Now you said it has a nucleus,
Yeah, every cell has a nucleus. It's human cells.
and in the nucleus, is that that's where the DNA is at, right?
Exactly, yes. Yes.
Okay.
So here it is, one or more, smallest, basic. It doesn't have to be smallest, it can be basic depending on the question. Smallest and basic unit. It comes from pre-existing cells. Okay, can you guys answer this for me? Living cells have an outer plasma membrane, cytoplasm, inside the membrane, and genetic material. What kind of genetic material?
DNA.
DNA, yes. Eukaryotic cells, remember it's eukaryotic, make up our complex organisms such as humans. The DNA in the eukaryotic cell is included in
Nucleus?
nucleus. Very good. Which cells, such as bacteria, have DNA with no nucleus?
Prokaryotic.
Prokaryotic, yes. The what to what ratio limit cells size, and that's why we have small cells. Ratio between what and what?
Surface to volume?
Surface to volume. Excellent. Yes. Okay. Now we are going to go to the different parts of the cells. Okay. And this is very, very important part. You have to know what I'm going to focus on. And we're going to start with the plasma membrane. And later, if you see plasma wall, remember that we do not have plasma walls. Humans do not have plasma walls. Eukaryotic cells, not just humans. Eukaryotic cells, no wall. Wall is in other things like bacteria. We don't have a wall. We have a membrane. Is it different? Yes. We are going to talk about the membrane, which is important for us. The membrane is 2 layers of phospholipids. Very important. You have to remember this word. Double layer of phospholipid or phospholipids bilayer. What's a phospholipid? Cut it into 2 pieces. Phospho and lipid. Lipid is like fat. Phospho is phosphate. So what is it? Phosphate and fat put together. Why does it have to be like this? I want you to understand everything as we go. Why? Why is it phospholipid? Why not lipid? I will tell you. The inside, remember, inside the membrane is called cytoplasm. The fluid part of it is called cytosol. I hope you guys are following with me. The cytosol is watery. It's watery. How about if this is a cell and there is another cell out here? What's outside of these cells, which is in between those? It's also watery fluid, meaning 2 sides of the membrane is watery fluid. You're facing water from inside, you're facing water from outside. Do we know hydrophilic and hydrophobic? What does it mean? Do you guys know? Hydro means water. Philic means love. Phobic means hate. Again, prefixes and suffixes. Collect them, you'll be good. Hydrophilic, love water. We also call it polar. And hydrophobic, hate water. What do you mean love water and hate water? Here's what I mean. If I give you some sugar and you put it in the water, it will dissolve. This is hydrophilic. It loves water. If I give you some oil or margarine or something and I ask you to dissolve it, it's not going to dissolve. It hates water. It's hydrophobic. So fat is hydrophobic. Everything else, hydrophilic. Okay? So what's the point that you're trying to make here? Here is the point. This membrane here is fat. If it is only fat, it will not be able to interact with the inside, which is watery, or outside, which is also watery. You will not be able to do that. You cannot interact with it. So in order to avoid this issue, but you have to interact with it. Otherwise, you will not be able to communicate. Remember, this membrane is selectively permeable. And this is a word that I want you to remember. Selectively permeable. We'll talk more about it. But for now, selectively permeable. What does it mean? Permeable means permission. Selective means not everything. Not everything outside of the cells can go in. Not everything inside can go out. If that's the case, cells will die. It's not the case. Selectively permeable. It selects who take the permission to leave, who is permitted to go in. In order to be selectively permeable, we put fat here. But you need to interact with the outside and inside, which is watery. How to solve this issue? I will solve it by adding phosphate to the fat. Look at this. If you look at the inside here, you will see these spherical structures, which is called the head, and you will see 2 legs, right? There's head and tail, okay? The heads is the phosphate ones, and the tail is lipid. So by doing this, you have the phosphate facing out. It can interact with the water outside. Look at the other side. It's like upside down. So the heads are still the other heads. It's 2 layers of phospholipid. Remember? Look at this. Phospholipid, 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 phospholipid. This is 1 layer. Here again, phospholipid, 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 another layer. So those are 2 layers of phospholipid. What's the arrangement? Phospho is a hydrophilic one. I told you, anything that's not fat, anything that's not lipid is hydrophilic. It loves water. It interacts with water. So I put the phosphate facing in of 1 layer. I put the phosphate of the other layer facing out. So I solved the issue. How about the tail? The tail will be in the middle with no interaction with outside and inside. And now I solved the issue. Look at at all of these individual phospholipids. Look, head and 2 tails. The head is phosphate, which is hydrophilic. Polar. Hydrophilic is called polar. You need to remember this. Hydrophilic is called polar. I'm not going to go deeper now, but you will understand it more later. But for now, we call it polar. The head is phosphate. So it is hydrophilic. What's hydrophilic? Hydro is water, philic is loving. It loves water. So you solve the issue. And the tail will stay inside to act as a barrier. So not everything will be allowed to go in and out. So everything that I talked about here is important. Again, feel free to stop me anytime you want. The plasma membrane is extremely thin. It's extremely thin enough that like if you stack them together, 1,000 will be like a sheet of paper. This is how thin it is. And it has to be like that in order to allow things to cross it. Everything? No, not everything. I told you selectively permeable. Things that selective things, certain things will be allowed to go in and out, and you will see that. Remember, there is fluid inside and there is fluid outside. This fluid is watery. It's liquid. So from outside, crossing to the inside is what you need, the good stuff. And the bad stuff that you created from consuming the good things, like nutrients. Nutrients is outside. You move it in, crossing the membrane. You use it. You get waste products, you kick it out. So nutrients is going in, waste product is going out. Across what? Across the double layer or the bilayer of phospholipids. Is it completely phospholipids? No, I'm telling you the basic structure. The basic structure, the building units, it's phospholipids. But we do have a lot of other things, most important of which is protein. You have the protein for different reasons. It's embedded within that phospholipid. Like if I ask you this question, let me just make it more clear. Your house, there is a wall of the house. Is your house completely surrounded by a wall, or there are in certain parts, there are windows and there are doors? There are windows and doors. The bricks that make the wall of your house, this is the phospholipid. The doors and windows and others, this is the protein within the wall. Is that it? No, we have a lot of other things. I'm telling you the most important 2. Phospholipid is number 1. This is the main one. This is the major part. This is the main structure in which there are some proteins. And the movement here is the word again, because it's important, that this membrane is selectively permeable. Not everything goes in, not everything goes out. It let certain things, select certain things are permitted to go in and certain things go out. Now, why do you have proteins? What's the function of this protein? Why do you have it? Remember, you cannot have the whole thing phospholipids. Nothing will move in and out. This cannot work. But you have to have certain proteins. Why do you have proteins? Some of them will work as enzymes, which is catalysts. Reactions the chemical reactions that happen outside and inside of your cells. In order to speed them up, they need a helper. This helper is a catalyst called enzymes. Enzymes are protein in nature. Others will work as channels. Remember, I told you the outside of your house is built from building blocks like bricks, or yes, it's basically bricks. You have bricks. These bricks, each one of those bricks is a phospholipid unit, right? But is the whole house just a wall? How can you enter your house? Right? No, you have doors, you have windows, you have other openings. This is what we're talking about right now, which is the proteins. Like you have the door of your house in order to go in and out. You have the window for the air to go in and out and to look outside and so on. Another opening, this opening is for the air condition, this opening is for the heater and so on. You have different things. It's not just bricks. So here, applying the same example, you have the protein that having different functions that you need to know. Number one, enzymes are proteins, which is a catalyst speeding up the metabolic reactions. Other proteins are channels. Things can move in and out, like water channels. Water can move in and out. Can water move in and out through the phospholipid? No, it cannot. It cannot. Why? Because look at this bag. You already know what's inside here, the tails. The tails are fat. If the water is coming here, okay, it will interact with the phosphate part, which is good, but it cannot cross. How does it cross? From the channels. Like, how can you cross your the wall of your house? From the door. Here you have it from channels. Other proteins will act as transporters. What's a transporters? Transporter to transport things from inside and outside. Why don't you use the channels? Not everything is small enough to fit in. Other things need something to transport it and take it literally from outside, push it in. So this is another one. Third one. You have a question?
Yeah, are the channels inside the lipids or inside the tails and the transporters?
Inside a protein which is embedded within the phospholipids, in between the phospholipids.
So it's in between the two tails?
It's between the units. It's embedded in the units of the. No, it's not. It have nothing to do with the phospholipid itself. The protein is embedded within the phospholipid. So around the protein is phospholipid, not inside of it. Inside of it, there is a channel.
Okay, I understand. Thank you.
Just like the door. Like, do you have bricks in the door? Right? You don't have bricks in the door, right? The door itself is in the wall. But how about the door itself? Yes, the door is a door. You can go in and out. Here are the channels. Where are the channels? Within the protein. Where is the protein? Embedded within the wall. So you're not interacting at all with the lipid part, which is a tail. Transporters is number two. Number three, receptors. Receptors are like ducts outside in order to respond to different things. I always say the receptors is like the ring outside of your house. Like when somebody come to your house, they push the ring, right? If you guys have that ring that have the camera that you can see, right? If somebody coming to you, does he just enter your house? Of course not. He pushed the ring and you say, yes, how can I help you? I have mail for you. I have delivery for you. Okay, I will allow you in or out, right? So this is the receptors. When you wanted to communicate with the cells, you go to the membrane and you knock on the door or you push the ring, and the ring is like the receptor. Like if you apply that, like if you have mail delivery, they come to you, they push the ring, and you communicate with them. The ring is the receptor. The mailman is the hormone. This is exactly An example of that. The hormone is like the mailman. He's bringing something to you. So the hormones go to the cells with a certain message that we will talk about later. So the hormone coming with a message, go to the receptors. The receptors will say, okay, yes, what do you need? I have an order for you from one of the glands, and the glands are telling you that you have to make energy, for example. Okay, thank you. I'm going to tell the inside. The receptor will tell the inside, the nucleus. And the receptor will tell the nucleus, there is a hormone that came here and it interacted with me and told me that this is what we have to do. And this is how it works. The other thing is recognition. Another protein. All of these are proteins, enzymes, channels, transporters, receptors, recognition, like a marker. This is also a protein. And this is very specific, just like your fingerprint. And here's what it means. Same example. If you have a mail delivery, the mailman is driving his car and he's having something for you. How does he know that this is your house and this is the house that I need? There is a sign on your house with a number in your street, like this is 23555 4th Street, for example. This sign with a number is the recognition sign, which is also a protein, and it is unique. So you cannot have two houses in the same street with the same number. It's impossible, right? It's unique, just like a fingerprint. And this is how our immune response or our immunity inside our body know the cells from each other. Like if we look at the cells, we can know them. This is fat cell. Yes, you're looking under the microscope and you have an eye, but your body does not have an eye and he doesn't know how to know. When I look at this protein specifically, which is the recognition protein, I know who you are, meaning red blood cells have a protein that recognizes them. When I see it, I read it, I know this is red blood cells. Liver cells have their own recognition protein. Muscle cells and skin cells and every type of cells in your body, they have a unique, like fingerprint-like identifier or recognition protein. So here it is, just to make it more obvious. Look at this. This is the main membrane itself. Look at this. Here is one layer out here. Here is another layer in here. Two layers. And notice that the heads are outside and the heads are inside. And what's in between are the tails of the two layers to act as a barrier. Look at this transporter protein. Look how it's crossing. So this is what you were asking me about. When something is moving within this, it will not interact at all with the phospholipids. It will go within this protein. Or if this is a transporter, the transporter will take it from outside to the inside without having to face or interact with the lipid phospholayer, the phospholipid bilayer. This is an example of an enzyme. It's going to catalyze reactions, metabolic reactions. What do you mean catalyze? Speed up the reactions. Here is another one that's a pump protein, pump. Things can move in, things can move out through this pump. Same for the channels. I told you about the recognition protein. Here it is. Look at this. You know what's going to happen here? If something is coming along this cell, he cannot see the whole entire cell. He just look at this. This is exactly like the sign outside of your house that's telling this, who are you? So when I look at this, this is unique. What you're looking at here, this is unique, like a fingerprint. Once I see this, let me see here. Oh, this is a red blood cell. Another one for the liver cells, it looks different, but same idea. This is a receptor. What's a receptor? Reception. Receptor, like ring. If something come, It does not cross. Actually, some hormones can cross, but I don't want to go deeper and distract you guys. Some hormones can cross actually if it's lipid, but others cannot. Yes, Kurt, go ahead.
So does each organ have like a different, because you said something about the liver cell. So does each organ have different types of cells just to kind of like identify which one's what?
Yes, here it is. Yes, each organ is mainly, not 100%, mainly made of certain cells. And these cells will have their unique receptor. A lot of these organs will have mainly one type of cell, but also additionally other types of cells. Example, stomach.
Okay.
What kind of cells do we have in the stomach? In the middle, you have muscle cells, and these muscle cells have their own receptors. Is that it? No. There is some mucus cells outside. Is that it? No. There are also gland cells. All of this in the nucleus.
Wow.
You got it?
Yeah, that's a lot. That's a lot to take in.
Okay. So just for now, let me say it this way. Each type has its own receptor. Red blood cells have the receptor. That's different than liver cells receptor. That's different than scale cells receptor. Let's take it this way, and then we are going to expand later. But for now, what I want you to know is what's the function of the protein, or what different types of proteins. Enzymes, here it is. Catalyst can act as a transporter, transport things from outside inside, or the other way around. Can act as a pump or a channel. Things can go through it without interacting with the membrane. Some of them are recognition to tell us what type of cell is this. You don't have to expand, and that's why I'm not trying to expand. I don't want to go beyond what we need to know. So if you know, just know that these are the functions of the proteins, and what does it mean? That's more than enough. So this is recognition protein, like a fingerprint, like a ring. I'm sorry, not the ring. I mean like the sign outside of your house. Receptor. If anything is coming, it go to the receptor so that it interact with the cells. Like hormone is coming. It's coming to this cell. Interact with the receptor. Who are you? I'm the hormone. Yes, why are you coming? Where are you coming from? I'm coming from one of the glands. I'm telling you this message. Thank you. You can leave. And the receptor will tell the cells what is it. This is for the protein. Is that it? Is it only protein? No, let me say it this way. The main bulk is 2 layers of phospholipid, like the wall of your house. It's mainly bricks. But then you have a window, you have a door. Here you have proteins. That's number 2. These are the main bulks. Number 1, the bulkiest, the largest, the main thing, phospholipid, 2 layers of phospholipid. What else? Number 2, proteins. We have proteins for different functions, 5 different functions. Is that it? No, there are also carbohydrates, but not as much as this. There are some carbohydrates in certain parts also for recognition and protection. Like it acts like something like this. So some of these will have carbohydrates. So here it is. This is the whole picture all together. Look at this. This is the cell membrane, same as cytoplasmic membrane, same thing. Look at this. Look at all this yellow. This is the main thing of the wall of the membrane, I mean, which is 2 layers of phospholipid. Here is 1 layer, head, tail. Here is the other layer, the inside one, head, tail. And remember, is the heads are at the periphery. The tails are inside. This is 1. What else do we have? Look at the blue. All these blue things, proteins, channels, recognition proteins, receptor proteins, and so on. What else do we have? Do you have some carbohydrates? Yes. Some of them are mixed with the fat. Some of them are mixed with proteins. The ones that are mixed with the fat or the lipids, we call it glycoprotein. I'm sorry, with the protein, call it glycoprotein. With the lipids, you call it glycolipids. What's glyco? Glyco is a prefix for glucose or carbohydrate. Glyco, carbohydrate. I know that I'm giving you a lot of information. You have to know it. Watch the video again, study it, and put together the prefixes and suffixes that I'm giving you. Here is one prefix and suffix. Glycolipid. What's glyco? Carbohydrates plus lipids. Carbohydrate is like the sugar. Glycolipids mixed together. Here it is for recognition. This is glycoprotein. What does it mean? The blue is protein, and this is glyco, which is carbohydrate. So you have glycoprotein. Other types of proteins work as integral proteins. Integral for integrity, to strengthen the wall. So this is the number 6 function. I already mentioned 5. Look at this again.
Can
Enzymes.
I ask you a question?
Sure.
Going back to the other picture,
Yes.
why is the glycoprotein, when you pointed to it, it's the same as the carbohydrate, the purple? Because I see
Yes.
the blue, the combination of the blue and the purple. Is that what makes the change in the name to the glycoprotein?
Yeah, this is a good question. Look at the blue alone. This is protein, all right? The purple alone is carbohydrate. This one, this example here is giving you both of them. So the purple is glyco, which is carbohydrate. The blue is protein, together glycoprotein.
Thank you.
You're welcome. So did you get it? What are the functions that I need to know for the proteins? Number 1, enzymes, catalysts, speed up the reaction. Number 2, channels. Number 3, transporters. Number 4, receptors. Number 5, recognition. Number 6, integrity, integral proteins. Does the integral protein have any one of the functions that we talked about? No, the name gave it away. Integrity, to strengthen the wall, to stabilize the wall. Okay, these glycolipids right here, this is to maintain the stability. This is a function, and help in recognition with the immune response. What's the immune response? Again, I don't want to expand beyond what we need to know. Like the white blood cells, when they come to the cells, they should not attack your own cells. They will recognize you. Some proteins are mixed with carbohydrate. We call it glycoprotein, as I mentioned here. And it also acts for transport and transmission. Let me ask you this, see if you're following. The bilayer, is it lipid or water? Lipid bilayer or water bilayer?
Lipid.
Lipid. To be more specific, phospholipid. I didn't ask about phospho here, but phospholipid is lipid. It's type of lipid. Okay. What is a mix of lipids, cholesterol, carbohydrates, lipids, protein? Yes, the last one is protein. Still talking about the membrane. Is the quality here rigid or fluid quality?
Fluid quality?
It is fluid. If it is rigid, it will be broken down and it cannot allow the exchange. Is it protein or lipid that carry out most of the membrane's function? Not the main bulk. I'm talking about the function.
Proteins.
Proteins, yes. Lipids is just the main bulk. But what's doing the work? Proteins. Proteins are the channels. Proteins are the enzymes. Proteins are the recognition. Proteins are the receptor. All these functions from functional proteins, from the size, the bulk, lipids. I hope that makes sense to you guys, and you should remember it. Selectively or semi-permeable.
Selectively.
Selective. It's selectively permeable. It selects what go through and what does not go through, what's allowed in and what's allowed out. And so here it is. These are the answers. And the other parts of the eukaryotic cell that we will talk about, remember, there is a nucleus here, there is a cell membrane outside, and we cover the cell membrane in details. There will be nucleus and the other organelles that we will talk about. Nucleus. And I will do the nucleus and I'll stop here. Nucleus is, again, the manager of the cell. I always say it like this. This is the master control, the manager. Because of that, the DNA, which manages the whole entire cell, is located within the office. What's the office? This nucleus, I call it an office. It's not a real office. I call it an office because I'm telling you that the way I like to imagine this is like a factory. The factory has a membrane from outside, the wall outside. The factory have a manager's office, and the manager is sitting within that office. And then you have different workers and different machines in this factory. So we're talking about the office manager or the manager's office. The manager's office is this nucleus. Who is the manager? DNA inside this office. So it is isolated. It's within this office. It is isolated and surrounded by its own membrane called the nuclear membrane. The nuclear membrane has openings so that the manager can go in and out. I'm sorry, I'm take it back. Not the manager. He can send messages in and out, not the manager itself. By the way, DNA cannot leave the nucleus. The pores, the openings here are too tiny. He can send only messages, but he need to sit down in the office, never leave. You can send messages. Look at this. Here is the manager, the DNA, sitting within this office, surrounded by the membrane called nuclear membrane. This nuclear membrane is also selectively permeable. Selectively permeable. Just like the cell membrane from outside, just like this. Yes, the membrane here is selectively permeable. The nuclear membrane is also selectively permeable. Okay. And who is the manager inside this nucleus? The DNA. Is it just DNA? No, it's DNA plus protein. This is how it exists, not just DNA. DNA with protein. And we call that chromatin. Chromatin. There is nucleus inside the nucleus. Nucleus in the nucleus. Like the office manager, there is a tiny office inside of it, like a storage or something. And they call that nucleolus. What's the difference between that and nucleus? You see the difference? Nucleus here, nucleus, and this is nucleolus. Okay, so this is extra. Nucleolus, it's not the same. Nucleus versus nucleolus. What's a nucleolus? Nucleus of the nucleus. Nucleus of the nucleus. Tiny nucleus. Why do we have this? Why do you have this additional thing? Does it have a function inside the nucleus? You told us that the DNA is the manager. It's managing the whole entire cell. And he's sitting here in the office. Why do you have this one inside here? This is additional thing. Yes, it is additional thing. This is to make something called ribosomes. You will be asked about all these functions. What's the function of the DNA? Carry the instructions to manage the entire cell. Is the DNA all by itself? No, it's combined with protein. We call it chromatin. What do we have inside this nucleus? Nucleolus. This is an additional one. Why? It makes ribosomes. What other ribosomes? We will talk about it, not right now. Just remember, this is where we make the ribosomes. We will talk about the ribosomes later on. Also look at these openings, all these openings. The space inside the nucleus is called nucleoplasm. Remember, look at this. What is this space between the nucleus and the wall? Cytoplasm, the plasma of the cells. How about this? Nucleoplasm, the plasma of the nucleus, the space. And here we have pores. These pores are made of proteins, and it is tiny. The DNA cannot leave. Why the DNA cannot leave? Because the pores are too tiny, too small for it. An elephant cannot pass through a tiny door. So this is like an elephant, and this is like a door that can, like a dog or a cat or something can go through it. So DNA can send a messenger. It cannot leave. Why is it made like this? Why the pores are too small just to allow the message? Because DNA is too important and too expensive that you will not allow it to go in or out, because it will be lost. If it's lost, the cells will die. Okay, so I'm going to stop here in the nucleus.